Hello, my friends. We're going to talk a little bit about proteins today, and hopefully you have taken your protein pre-test before you watch this, because I need that information for Brighton. So thank you very much for doing that. That is awesome. Let's take a look at proteins, how they're structured, and what's going on. This happens to be hemoglobin right here. This is Ribisco protein right here. This is collagen protein right here. And these are particular enzymes that are used. So they've got these, proteins have these specific shapes. And they have lots of different, so, sorry about the sound effects here. We talked already about enzymes. Enzymes are proteins. Not all proteins are enzymes, but pretty much all enzymes are proteins. And we've talked about cell communication proteins. We've talked about those. We've talked a little bit about aquaporins. Those were in the embedded in the membrane of cells to let water go back and forth on either side in the cell membrane. Haven't talked a whole lot about hemoglobin yet. Haven't talked too much about keratin and collagen structural proteins. We've talked a lot about these ligands and the receptor proteins. We haven't talked a whole lot or at all about antibodies or actin and myosin moving or proteins in storage. But we will. So let's take a look at how proteins are made. The building blocks of proteins are just strings of amino acids, and their structures of the amino acids are coded for by DNA. You probably knew that already. So what does your DNA do? It codes for amino acids in a particular order, and the order of the amino acids determines the type of protein that's being made and the shape of the protein that's being made. And so a monomer is what's called a building block of a molecule. So the monomer of a carbohydrate is a glucose molecule. Monomer of a protein is an amino acid. So these are the building blocks that make up the molecule. Polypeptide is a string of amino acids together. It's a chain. And when that chain of amino acids links up and folds up all together, it's going to form a 3D structure. Let's take a look at what makes up an amino acid. There are 22 of them. What makes them up? You might have heard about an R group or a side chain in chemistry, this right here. But essentially, the rest of the molecule is the same. So this main part of the molecule of an amino acid has an amino group, this carbon backbone, and then a carboxyl group on the other end. This part right here, the R part of the molecule, is the rest of the molecule, and it's what makes each amino acid different. So what happens with those R groups? They determine the specific characteristics of the amino acid, mainly whether it's hydrophilic or hydrophobic, and then polar and nonpolar. So if I'm nonpolar, I'm hydrophobic. That means I don't like water. So some of the amino acids don't like water, which is going to be important to us in a little bit as we talk about protein structure, because that determines how they fold up. Those little chains of amino acids all linked together, how they fold up and make their certain shape is determined whether they're hydrophilic or hydrophobic. So you've got some that are hydrophobic, you've got some that are hydrophilic based on those R groups, whether they dissolve in water or not. You also have amino acids that either have an acidic side chain or a basic side chain. And they're either going to accept or donate hydrogen ions. So if I'm acidic, I'm going to release hydrogen ions. If I'm basic, I'm going to accept hydrogen ions. So that's another property of amino acids as to whether they have an acid or a base side chain. And that'll determine how they also react when they're in an aquatic environment. Amino acids have these disulfide bridges that are holding them together. And these are some of the bonds that can be broken with high salinity, low salinity, high temperature, or acidic environment, things like that. These little bridges that are holding these things together. So how do we get two amino acids to link up to another to make that polypeptide chain in the first place? Well, here's one amino acid, and it's got an OH at the end. This is a carboxyl end. And then it's got the amino end of another one that has a hydrogen at the end. So here's my NH2 at this end, and here's my COOH at the end of this other molecule. So they're opposite ends together. And what we're going to do is a process called dehydration synthesis, where we're going to remove a water molecule, H2O. So I'm going to remove the OH group from this amino acid and the H from this amino acid, and pull a water molecule out. And now these two right here are going to form a peptide bond in between the two amino acids. So here's this end of this molecule. Here's this end of this amino acid molecule. And now when I've removed my water molecule out of there, I form a peptide bond to put them together, called dehydration synthesis. And there we go. So the amino acids are linked together going from one direction. They are not added to on both sides at the same time. They're only going to grow in one direction. They're going to go from the N terminus, or the amino acid, the amino end, to the carboxyl end. Big thing about proteins is that their shape determines what they do. Shape determines function in proteins. So we looked at that at enzyme, the enzyme specifically catalase, and how the shape of the catalase is going to fit with the substrate. Well, if a protein's shape is altered in some way, shape, or form through some environmental factor or mistaken DNA or what have you, it's going to mess with the function of the protein. We're going to see what happens when there's a mutation in a particular protein of hemoglobin that will cause red blood cells to have a different shape because their proteins act in a different way. We'll talk about that in a second. So the primary structure of a protein is simply the amino acids in a certain order in a chain. So it's one-dimensional. It's just a line of amino acids strung together based on the DNA code. If I take just one amino acid and change it, it could change the entire structure of the protein. Let me move this down a little bit so we can see here at the bottom. There we go. We can completely change the protein, or we could not change it at all, or we could slightly change how it works depending on which amino acid. So arginine, asparagine, threonine, asparagine, glycine, serine, threonine. If I took any one of these and changed it for another one, I could either drastically change it or not change the function of the protein at all, because maybe that amino acid that I'm switching out for it acts the same. Maybe it's a hydrophilic exchange for a hydrophilic. But if I take a hydrophilic molecule or amino acid and I exchange it for a hydrophobic amino acid, that's going to change how the protein folds up. Let's take a look at an example of that. So here's an example, a famous example. It's sickle cell anemia. And you can see these beautiful red blood cells right here. They're nice and round. The hemoglobin on them is fine. So valine, histine, leucine, threonine, proline, glutamine, glutamine. That's the order of those amino acids. In this one, the mutated form, my one glutamine right here has been exchanged for a valine. Valine happens to be hydrophobic, and glutamine is hydrophilic. So if I'm hydrophilic, I like water. If I'm hydrophobic, I don't like water. So what causes the shape of this is the hydrophobic valine amino acid causes the protein to want to go away from the water, which is all around the cells, right? There's all kinds of water around red blood cells. And so it causes that protein to want to go away, or the amino acid to go away from that aqueous environment and that causes the funky shape of the red blood cell, which causes then sickle cell anemia, which we talked a little bit about in evolution, right? So that's one amino acid difference makes a huge difference in the structure, hydrophilic versus hydrophobic. All right. So the secondary structure, now I've got my amino acids strung together in a chain. And before I can fold them up into their complete 3D structure, I have to have some what's called local folding. And then I could have either the sheeting or a helix formed. So this is what's called a beta sheet form, where I've got my long string of amino acids together, and then they kind of kink up like that. Kink, 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 kink. That's called beta pleating. Or they can form like a helical structure, like a spiral staircase. They can form like a coil shape. So they're going to do some what's called local folding. And we have some attachments there. Our tertiary structure is, okay, we've got some local folding that happened. We had some pleating or some spiraling. Now we're going to fold that whole protein chain in and on itself based on the hydrophilic, hydrophobic interactions between amino acids. So you would have the hydrophobic amino acids that are going to fold inward to the middle of the protein, so they're not around the water environment. Your hydrophilic amino acids are going to want to stay on the outside of the folding. So here may be some hydrophobic amino acids. They're going to fold inward, so they're protected away from the water environment, while my hydrophilic molecules are on the outside of this folded protein up there. Our quaternary structure only occurs when we have two or more proteins that are going to work together. For example, this is hemoglobin, and there are 1, 2, 3, 4 proteins that make up a hemoglobin molecule. So quaternary structure only happens when there are two or more protein chains, polypeptide chains, that are going to work together. Not all proteins have a quaternary structure. All proteins have a tertiary structure because you have that 3D effect where it folds it on itself. But some proteins are going to work with other proteins and form this what's called a quaternary structure. Collagen is an example, hemoglobin is an example of proteins that have quaternary structure. So primary structure, you have your chain of amino acids. Secondary structure, got some local folding that happens based on the R groups. Tertiary structures when it folds it on itself, and the hydrophobic goes inside, the hydrophilic stays on the outside. We're going to form some bonds there. And then our quaternary structure is when we have multiple polypeptides working together. So we talked a little bit about this in our enzyme lab. What happens if these bonds that are holding this protein, this mature protein that's been formed, in its particular shape? So what happens? We can have temperature, pH, salinity, all these things are going to break the bonds. So here's my protein here, my three-dimensional protein. What happens to my shape if I break some of the bonds holding it in this shape is it can kind of unravel. Once it's unraveled, it forms a different shape. The different shape is probably going to cause it to have a different function. So when we denature proteins, we tend to assume that they're not going to function properly, which is typically the case. When you break those bonds, the protein changes shape, and then it doesn't have the same function anymore. So there you go. All right, and that is proteins, my friends. Brief little overview for you, and I'll have some questions for you to work on. Your proteins POGIL that'll take you into a little bit more detail, but with some pictures for you to take a look at. So, thanks for stopping by.